Does everybody have a handout? Nice. Okay, really good. Well, we've got 8:30, so why don't we get started? Good morning, S&P. Hey, that's how we always start. We have lots of visitors today. We always start out with a really good welcome to make sure everybody is oriented. I wonder if we can just have visitors say their first name and where they're from.
Hi, I'm Marcel, and I'm from Michigan.
Christian.
Yep.
I didn't know how we were going.
Please go ahead.
Okay, I'm Katie. I'm Catherine, but please call me Katie.
And where are you from?
Thornville, Ohio. 30 minutes down the road.
Okay, great.
I'm Christian. I'm from Grosse Pointe, Michigan.
Okay.
I'm Steve. I'm from Philadelphia, Pennsylvania.
All right, very good, very good. All right, and we have another visitor coming in? A couple more visitors coming in. Oh, wonderful. Please come on in and join us. You are whom?
Craig.
Craig, and where are you from?
Westerville.
Okay, real good. Please join us. I'm glad that you're here. So for our visitors, this class is Sensation and Perception. We have in the morning 2 psychology classes going on this semester. Both of them are upper level, but we do try to make them accessible to people who might be joining us. So this class called Sensation and Perception is a little bit on the biology side of psychology, and that works for those students who might have an interest in neuroscience. And we'll be talking today about something that our students have read for today's session. It was a work where some recordings were made in the occipital lobes and other portions of the brain in monkeys out at MIT when they were performing different kinds of tasks. So we'll be talking about that today, and we'll have our students do an exercise also in just a little bit. For the students who are in this class, I thought I'd just orient us a little bit. Please make sure that if you're a student in the class, you do have the writing assignment that will be due not in 1 week, but a little bit more than 1 week. So here we are on October 28th. And we'll be talking about this article in just a few minutes. What I did want to give you a heads up about is that writing assignment will be due all the way out here on Friday the 8th, but now you have plenty of time to work on that. And I thought I'd also give you a heads up about our good news, which is that we have an exam on Wednesday. So let's give it up for the exam. Yay! Our students are always excited about the exams that they're going to have. And we've already done this particular PowerPoint, but just to make sure all the students are on board with this, here's the format of the exam. As always, this is the same format that we've had many times. Usually our exams here are short answer kinds of exams. And the content is spelled out here. Nothing particularly new, except we're going to remind you that we will emphasize the material since October 14th. Okay, and that's when we had our last exam. So our material here is cumulative, but we do try to emphasize what we've done most recently. Here are some tips on how to practice, and this is available to you out on Blackboard and out on the S Drive. Any questions from our regular students on the exam? Okay, all right, that all sounds really good. I wonder if we can take a moment and go over the writing assignment, and to do that, we'll make sure first that everybody has one of these if you're a student in the class. Then we'll ask somebody if they wouldn't mind reading from the top, and we'll go just down to the bottom of number 4 here, so we can make sure that we're all on track. Do we have a volunteer reader for us? Thank you, June. Nice and loud so people in the back can hear you, please.
I'll just start with the number.
Why don't we start with, yeah, okay, that's fine. Okay. Number 1?
Subsequently, write an essay that addresses each of the following. Number 1. Using approximately 250 words, form your best argument supporting Sharon and Candy's decision to seek a deaf sperm donor. Number 2. Using approximately 250 words, form your best argument for why Sharon and Candy should have instead sought a hearing sperm donor. Using approximately 250 words, form your best argument supporting the
Gallaudet is the school. Yeah.
Students' demands for the resignation of their hearing president, Dr. Elizabeth Zinsler. Number 4. Using approximately 250 words, form your best argument against the Gallaudet students' demand for the resignation of their hearing president, Dr. Elizabeth Zinsler.
Okay, so thank you, Jim. We have just a little bit of context here, so probably our visiting students have no idea about what's going on here. One of the writing assignments that we'll have, we'll try to connect something in the sensory world, something that we're learning about in class. Right now we're dealing with the visual system, but in the next few days we're going to move on to the auditory system. And we're going to be reading about a couple that happens to be lesbian, and they happen to be deaf. And they wanted to find a deaf sperm donor for their child. And so you can imagine that this might be somewhat controversial, and the students will be writing about that. We'll also connect this to a school called Gallaudet. How many of us have heard of Gallaudet before? What do we know about Gallaudet?
It was like one of the first deaf schools.
Yeah, great. Okay, it's actually a predominantly deaf school, and for the first 100 years or so of that school's history, they had presidents who were hearing. And in late 1980s or thereabouts, there was a movement among the students to try to get a deaf president, and I think they've largely had deaf presidents ever since. So there's a wiki on that, and then there's also several videos on that. And the one that I think I'll have you watch, and we won't do the whole thing right now, it's a video on YouTube that is entirely free of sound, and you'll see in just a moment some captions start rolling up. And we won't go all the way through it, but this is the Deaf President Now video that we'd like you to comment on, and this is available to you by some of the Blackboard links that we have. But you'll see in just a moment the news story starts, and we have the captions here, which might be a little bit difficult to read under this lighting condition, but that'll be the basis for your essay. Okay, so I think it'll be really quite an interesting conversation that we'll have on that Friday. I'm looking forward to reading your papers already. Any questions about that assignment or how it connects to sensory systems? Okay, should be really, really good. All right, let's go on then and remind ourselves of what we were reading for today. Today, the students were reading an article that came from MIT. Here it is. And this is entitled Top-Down versus Bottom-Up Control of Attention in the Prefrontal and Posterior-Parietal Cortices. So just to get us all oriented, why don't we all point to our frontal lobes, and our students can join us too, our new students, if you know where our frontal lobes are. Okay, our frontal lobes. How about our parietal lobes? Okay, so we're going maybe up top here for our parietal lobe. All right, really good. So why don't we start out this way? Why don't we, I've done this several times over several semesters with this paper, why don't we have our students spend about 5 minutes jotting down everything that you did understand about this fairly technical paper. Okay, so take a moment to refresh your memory about this. You can make bullet lists, you can write out sentences. I won't be collecting these, but we want to have you refresh your memories for what you got out of this paper. While you're doing that, I'll chat with our visiting students just a little bit. So who has an interest predominantly in psychology among our visiting students? Okay, and who has an interest predominantly in neuroscience? Okay, so we have, anybody have a combination of both of those? You're interested in? Okay. Okay, that aren't math. Okay, all right. Okay, so you almost wound up in a math class over there. Okay, all right. Okay, yeah, I was a little surprised when you said that you were here to watch.
I misread the thing, I guess.
Okay, all right. Well, we're glad that you're here. Okay, who's taken some psychology courses in high school? And what kinds of things have you done there? Anybody want to share with us what you've done?
We just finished like our unit on the brain, like on the lobes and everything.
Okay, okay, so that was an easy question for you, right? Okay, something else? What have other people done? Maybe in an AP course, if you've or whatever kind of psych course you might have had.
We used to have one within the person that teaches and moves to guidance.
Okay.
So then, the way I guided like stuff about that is by reading myself.
Okay.
Different things. So
Uh-huh.
I wish I could have taken it because I was on college credit, but she moved.
Okay.
So
Okay. All right. Feel good. In a few moments, what I wasn't sure whether we'd have any visitors here or not, and so what I was planning on doing is having this group actually run an experiment. I'm moderately inclined to have you run the experiment. We're going to take you upstairs. It's a computer-based experiment, and we'll have you do the behavioral tasks that the monkeys would have done. I was going to have our students do that, but maybe you would enjoy doing it. It takes about 10 minutes or so, and it might give you an appreciation for what goes on here. Maybe we can have the students here be the experimenters, and we'll have you be the monkeys. So I guess you all slept here last night, is that right? All right, how were the dorms? They worked? Yeah, okay. All right. Any questions about life in Granville? I also live in Granville. I've been teaching here since 2001, and so this is my 13th year. I actually taught my first class in this room. I've been teaching sensation and perception for the 13 years, but I've been living in Granville the whole time. So maybe you have questions about Granville? I know some of you are Ohioans anyway, and maybe you know Granville, or, yeah.
I live close here, but is the weather normally just like following 4 seasons? I live 30 minutes down the road, but things change in 30 minutes. So.
Yeah, it wouldn't be that different, right? It wouldn't be that different. It'd be a little bit warmer than it is in Michigan, but not a lot warmer than it is in Michigan. We get the same winters you get.
Okay.
Yeah.
Because sometimes it's like, it's different than the 70 and the road between it.
Yeah.
It can be very, very strange.
Okay. Also we had somebody visiting from Philadelphia. Is that right? Yeah. I'm a native New Jerseyan myself, and usually what happens is the weather patterns move from about here over to your way. Every once in a while you get a storm coming up the East Coast that we don't get. But generally speaking, the weather that we have today is stuff that's coming your way. So the weather wouldn't be too different for you either. Okay. Any other questions about psychology in general or neuroscience? Yeah, thanks.
When we can take the neuroscience concentration,
Uh-huh.
they biology, psychology, they pair together.
Yeah, so the courses I think are listed on your handout. They may or may not be, right? And so you always have to take Intro to Psych and you always have to take Intro to Bio. And then if you go that far and you still are interested, then you could take Intro to Neuro. And then the concentration is really a mixture of Psych and Neuro classes and a little bit of chemistry also. Not very much chemistry. Yeah. Okay, I think that's about 5 minutes or so. So here's what I was going to do. Now that you've gotten your ideas down on the page about this fairly technical article, this is in a very prestigious science journal, and of course it's MIT work, so you can imagine how technical this might have been. And yet our undergraduates are taking a shot at this. They're giving their best try at trying to understand this, and it is a little bit technical. So one of the ways that I think it's best for us to begin to understand is to really get a feel for what the behavioral task was. And in order to do that, I had set up upstairs, we have a lab that has 10 different computers. I set up something called Cog Lab. This is a piece of software that runs visual search tasks, fairly similar to the ones, not identical, but similar to the ones that were going on in this MIT paper. And some people who are in our research class are actually running the visual search, right? So I've got the same one queued up over there. And I was going to have the 10 of us or 13 of us go up and run that, minus the 3 students who are in that class. We do have 1, 2, 3, 4, 5, 6 new folks on board here. So I wonder if we might have them run, and then we could also have a few others. If you want, we have some space up there. If a few other students want to run that, that would be fun. I might ask if the people who are in the visual search group could help us run the Cog Lab upstairs. We have them loaded up. And then we'll see if we can collect a little bit of data. This might take us 10 minutes or so. We'll come down, and that might help clear the way, give us a little bit more of an intuition about what was going on in this study. Does that sound okay? I think that's the best way to handle the folks that I'm delighted to have visiting us today. Okay, so I'll play Pied Piper. If everybody will follow me on up to room 515, you can leave your stuff here. We'll be coming back down. And if you feel uncomfortable about that, I'll lock the door behind us. Or maybe I'll ask June, if June wouldn't mind. June, could you just close the door behind us so that nobody comes in? All right, and let's go this way. Our lab is in here, 515. And we'll get 1 student per room. I'll just sign onto the computer and we'll get you started in a moment. We'll get 1 student per room. If each of our visiting take a seat in the room, I'll just log us on. Pardon me? And if we have some of our visual search students, any of those folks around are visual search experts, okay? I wonder if we could have the students who are visual search experts begin helping us. So you can see Cog Lab is already loaded here. If somebody would step in, get this going, and I'll get our next student signed on. Pardon me. Yeah, I'll log on. As a matter of fact, okay. Can we get some other visual search students, who else do we have? Or any of the students in the research class, could you log on to any one of the computers? That will just, you can click on switch user if you want, and you can log on. Okay. You can find an open, click on user, log on. You ready to go? Yeah. Okay. We're going to click on visual is going to get lower here. And your target is going to be all kinds of distractions. If you see it, you can hit this. Yeah, and then you trial to escape. You can see it. Okay. All right. We'll see if we can get our students going. Okay, really good.
Can they just start whenever?
Yeah, please. The sooner the better. And your target is going to be a green circle. So if you explain that the target is the green circle embedded in the sectors. Okay. So, and what will happen is if you see the target is present, you can put in this key. And if you think that the target is absent, you can put in that key. Okay? And we'll start every trial with the spacebar. So you can click OK. Huh? All right, and spacebar to start. And we can use either that slash key. So that's going to be
slash if it's there.
you can think of it as being zip. It's not there. So zip is 0. It's not there. Okay. We'll hit the spacebar again. There it is. Right. Okay. We'll practice that. All right. Are all of our regular students now? Everybody's running? Okay. Great. Okay. Anybody else want to do it? So I know that we have some folks who are experts at this and wouldn't need to do it. Anybody else want to get a feel for you're invited to participate in this if you want? Yeah? Would you like to do it? Naomi, thank you. Right. If we play our cards right, we might be able to use some of this data as some baseline for the group. Right. So, Naomi, if you'll just sign yourself on. Okay. If you want to find an open terminal and sign yourself on? Okay. All right. That's fine. We'll make sure that we're moving along. It only takes about 5 or 8 minutes. Okay. We'll make sure. Okay. And so the target is going to be a green circle. Okay. And if you see the green circle, you can slash it out with that slash sign. Mm-hmm. And if it's not there, you can say zip. It wasn't there. Okay. All right. So here we go. And you'll start. Okay. And if it's not there, you can. Okay, right? And then you can press the space bar for the next time. Okay. Rolling. It'll be done in about 5 minutes. All right, so this is like the MIT lab. Everybody's doing visual search. We'll get this one going. Okay, so what will happen is circle, okay? And the green circle will be your target. You can slash out the target with that, okay? And if it's not there, you can say it wasn't there with a Z, okay? All right. And you can hit the space bar to start each trial. Get this out of your way, and that says zip, right? Because it's not there. And then you space bar to start the next trial. Here it comes. Okay, that's all right. And so you can space bar. All right, and we'll take. For our other students in the class, if you want to look over their shoulders and just see whether you would report present or absent on every trial, that's basically what they're doing. They're trying to find that green circle. You might be very familiar with it, so maybe you don't need to look. But if you just want to see what they're doing, green circle, present or absent, and there's 2 alternatives for this task. thing is that when they finish, we'll leave their data up. I'll swing by later and see if I can capture it and put it into a spreadsheet for that group that's working on that, right? It might give us some kind of a baseline to work with. It might be that we don't need it because I think you figured out you wanted to do this all within subjects.
Right.
Right, okay, so, but anyway, we have something.
Just in case.
Yep. Are you already done? Yeah. All right, wow, so fast. Okay, really good. Your data, okay, real good. Okay, that looks really, really good. We're going to leave that there, and then later on, we'll see if we can capture that and maybe get some kind of a summary for our next class. But anyway, we'll go down in just a few moments, and when the other class finish as well, and we'll have a conversation about how to get those data, and then what might be the neural correlates of all of that. Hey, we got somebody else who's done. Thank you. Okay, so here's your reaction time data. And sometimes you had 4 distractors on the screen, sometimes you had 34 distractors on the screen. Sometimes you had screen filled with everything, 64 distractors. Okay, and here you can see when you're slower and when you're faster. And for these darker items, the conjunction searches, we were quite a bit slower. Here it's like golf, lower scores are better. Okay, all right, so we'll just wait for a moment for the other folks to finish as well, then we'll head downstairs and we'll talk about how to interpret this. You're all done, okay, real good. I wonder if you can take a peek at your data tables. Data plot. Wow, okay, great. All right, why don't you come on out. We'll begin to head down in just a moment. Maybe we'll wait for the group to just finish up. You folks got a little bit later start.
I don't know what that means.
Good. This means we don't know what the
I hope it means the name.
well, everybody gets a similarly shaped graph, and we'll go downstairs in a moment and talk about that so we can understand what the behavior looks like, and then we'll see if we can understand something about the physiology.
That test for when you get your driver's license.
Okay.
You know, you have to, can you perceive this dot and
Yeah.
the other side of the screen?
Right, right.
Also have some laundry.
Okay, is that everybody? Are you all, okay. And did you take a look at your data just to see how your data look?
Couple of them.
That's okay, that's okay. Okay, is everybody done? No? Oh, sorry. Okay. Plugging away. Naomi's still plugging. Okay, just in the interest of time, why don't we move this group down? I'll stay here for a moment with the other group, and I have some sample data downstairs that we'll talk about, and then we'll do the ePhys, the electrophysiology on this stuff.
How do they appear on the screen? And I read red, yellow, green, and that was actually what I was.
I'll meet you down there in just a moment. I'll finish up with these students. All right, looks like my friend here, Christian, is done. Is that right? Okay, Christian, if I can show you what your data look like. These are your data, and your reaction times are better than mine. You're faster than I am on this task, okay? Here's your reaction time. Lower scores are better. This is kind of like golf or running, right? Lower scores. And this condition seems to have given you the most difficulty. This is called something conjunction absent, and we'll be talking about what that means in just a few moments. And then here's your conjunction present. And then these feature searches, you appear to be extremely fast. You're chiming in something like 530 milliseconds, okay? And here we have the different set sizes. Sometimes we'll get 4 on screen, and then it seems like this filled with 64. No, that was in between all of those two, right? So we'll see what your, how you look downstairs. Okay, Naomi, and we'll just have you take a quick peek at your own data. Your own data will look very much like mine. The dark items are conjunction searches, and those are relatively slow. Here's reaction time, and you're really fast on the feature searches. All right? So why don't you, Christian, and I head on downstairs? Yeah, there you go. Good morning, Frida. How you doing? Good, nice to see you. Okay, everybody. Thank you for participating in the hands-on experiment. I think we did that fairly well. We got over a computer glitch or two and we made our way there. Let me see if I can find my data. I ran through this earlier this morning on this computer. Here's CogLab, and here's my stuff. Okay, so first, I wonder if we can impose on any of our students who just took that, some of our visiting students. Can you describe briefly what that task was, what kinds of stimuli you saw, and what you had to do? Anybody want to begin to describe that for us, just to orient us? Okay, yeah, thanks.
It's kind of easy in the beginning, it was like all blue and then one little green thing, like pretty easy to go through, and then it got, oh yeah, there's more green going on. It got more and more, but
Okay, how many people had that experience? The people who ran in that, it started out really easy. There was one green among blues, and that was really easy to do, right? So you're trying to find a target that is a green circle, and it might be a green circle amidst blue squares. Really, really easy, right? Really easy to do that. And then all of a sudden, as you get into maybe 5, 10 trials or so, you have to find that green circle among green squares and blue squares, right? And that was a little bit more difficult. All right, so here is a sample of the data, and I think most of our students were faster than I was, so I'm not quite as fast as the students, but we show the same kind of a pattern. Now I wonder if I might impose on somebody who's in our regular class to begin to explain what some of that terminology means. And I'll turn down the front lights just so we can see it a little bit more clearly. Becca's got something for us. Thank you, Becca.
I'm not quite sure what the exact words are, but
Okay.
My guess is that feet absent and feet present are when you were looking for the green circle amongst other blue stimuli.
Okay.
And absent and present just refers to whether or not the target stimulus was actually there.
Okay. All right, really good. Can somebody else, maybe somebody who's in our Psych 341 class who actually might know what the CONJ stands for? Elise, if you wouldn't mind. Okay, thank you.
The CONJ stands for conjugate, which would be a search that requires selective attention. So one of the searches where it was harder to find the green circle
Okay.
would be a conjugate search. And the feature search is when the stimuli is very salient. So you can easily notice the green circle amongst just a few squares.
Right, so if it's one singular green target amidst all of these blues, that's something that we call a feature search, and that's actually something that we also call pop-out. It sounds like a very informal phrase, but very technical people use that phrase, and that phrase actually appears in our journal article. Pop-out is this feature kind of search. It's finding a green among blues. It takes almost no time to do that. Look how fast we are in these feature searches. Whether the target is present or absent, we get a relatively flat behavioral performance. That is, reaction times are relatively low. Lower scores are better for reaction time. And it's almost as if it doesn't really matter whether we have 4 distractors or 16 distractors or 64 distractors. We're really, really fast if we have to find one green among blues. Who's okay with that? Now when you have to start looking at the conjunction of features, and the conjunction might be that you've got now a green circle among green squares and blue squares or something like that, then pulling that conjunction out does take some time, and you can see that we're actually losing some time. This is the same pattern that we saw in all of the graphs upstairs, and it's particularly salient when we have something like 64 distractors. Okay? You really have to almost go bit by bit. Did anybody have that experience? Of the students who were running this, you felt that sometimes you really had to move your eyes around in order to get that? Did people have that experience? Yeah? Okay. Anything else you want to describe about that?
I just took it simply. I just scanned stuff, so like if I see anything that doesn't look like a square,
Okay.
get it, but if I didn't, I just
Okay. Okay. I think I saw this pattern was there for each of the folks. So that's the behavioral task, very similar to what our MIT monkeys were doing. So many thanks to our students who came in today to play MIT monkey for us. They didn't know they were doing that when they came in. Okay. So I wonder if we can just now have somebody else maybe walk us through what was going on here. Something very similar. This is the paper that we read for today. Similar kind of visual search, not exactly the same, but close enough perhaps. Anybody want to help us out with that? Harshita and then maybe Jenny.
They put electrodes in monkeys in 3 areas.
Okay.
2 in the frontal cortex and 1 in the parietal.
Okay.
It's called LIP and VFC and FCF.
Okay, right.
And they did recordings
Okay.
of firing rate, and a particular neuron found a stimulus when the firing rate shot up significantly.
Okay. Uh-huh.
That is
Okay, just before we get into some of the physiology, so thank you for that good lead. Can you walk us through just that behavioral task one more time?
They had 4 panels,
Okay.
I think, and one of the 4 would contain the target stimulus.
Okay.
And in the pop-out, the target stimulus was distinct from the 4. Other 3, whereas in the search one, all the differences were distributed, so it wasn't that serious.
Yeah.
You had to sort through.
Right, okay, so sorry if this is a little bit dark, but it starts out with this presentation, and what they're giving you is this green line. That becomes your target on this particular task. That green line might be tilted a little bit off of vertical, and then they're going to ask you to try to find that, and they're going to ask the monkey to make a eye movement to the quadrant on the screen. It can be upper left, upper right, lower left, or lower right. The monkey is fixating dead center in the screen, and they first show the monkey that here is the target that you need to find, and then they have the monkey go into a little delay period, and they're still having the monkey fixate right there, and then that target might appear in one of the 4 quadrants. They have to make an eye movement to it, okay? And they have these eye trackers. They know whether the monkey's making a correct response or not, just like you were getting feedback. How many people here saw that they were getting correct or incorrect feedback on, okay? And I know I made several errors this morning too, okay? So the monkey gets a juice reward for getting a correct response. Now sometimes, as Harshita correctly pointed out, we have the pop out. So here's that green vertical, and it's amidst differently colored items, and that's really easy. It's unique in orientation, and it's also unique in color. This is kind of like having that one green item among all those blue items. But then what you can do is you can have that same target now in a visual search, and you might have other greens that are there, but they're tilted the wrong way. You might have something that's tilted the right way, but it's the wrong color. What the monkey has to do is figure out which particular target is relevant based on the conjunction of features. And the conjunction of features might be something like color and orientation in this case. You could have used different features, but they're using color and orientation. Who's following that? Is that working for us? Anybody confused by what that stimulus is? So we have these 2 different kinds of tasks, which they call pop out, and they're calling it search. In our study upstairs, we call those equivalently feature and conjunction, but they're the same ideas. They're labeled differently, but they're the same ideas. Who's following that? All right, that's all working for us. Then Harshita had gotten us started really, really well. There were electrodes in 3 different places. Basically, though, there were 2 different places. There was relatively frontally placed electrodes, and then something that was a little bit more parietal. Within the frontal lobe, they had something that was sort of dead center above the eyes, and then something that was a little bit more lateral. But still, frontal-ish versus top-ish is where the electrodes were. Okay? All right. Anybody want to help us with what else was going on there? Let's go to the next page, maybe. There are some difficult graphs here. These are some really challenging graphs. Harshita gave us a good start, that they're looking at electrical activity while you're performing the conjunction search and the feature search. Okay, so here are some graphs. Anybody want to try to get us started on these?
Just a
Okay.
question.
Sure.
What does the term saccade mean?
Okay, really good. So what does the term saccade mean? So a saccade is an eye movement. Okay, a saccade is an eye movement. And that's really what happens in this study, is that the animal has to make an eye movement. In our study upstairs, the students were hitting either the slash key or the Z key. They didn't have the monkeys hit one of those 2 keys, although you can train a monkey to do that. They're actually fairly good at hitting selective keys. But instead they had the monkey make a different kind of motor response, rather than with fingers, with their eye movement. And they call that the saccade. So they're measuring the activity in the brain just before the monkey makes an eye movement and shortly after the monkey makes an eye movement. Okay? And the monkey's eye movement is given by this on this time axis, so time from saccade. Okay, leading up to the eye movement, and then a few tens of milliseconds after the eye movement. Okay. Okay. Anybody want to help us out with this? How many people were confused by this when you were reading this? Okay, this is pretty technical stuff, so why don't I help us out a little bit here. So what you could do is, and our folks in AP Psych might know about this, I have my stuffed animal neurons here. Okay, and the neurons begin firing, and we call these action potentials. How many of us have heard of those before? Action potential. Okay, great. A lot of us have heard of action potentials. One of the dependent variables that you can measure in psychology or in neuroscience would be the number of action potentials that you have. Another dependent variable that this group learned about many, many weeks ago, but it's been a while, it's called neural synchrony. And that is to say not just how many action potentials there are, but is this neuron firing synchronously with this neuron, or are they not synchronized? Okay? So that becomes a different kind of variable, and that's what we're measuring here in the MIT lab. As you're performing the task upstairs, was there any neural synchrony going on? Okay, and let's see if we can get some neural synchrony going here. We'll see if we can get maybe different rates of neural synchrony going on. I wonder if you can all tap with me in this pattern. So this is my group of neurons. Okay, you're all my neurons, and now you're firing synchronously. I'll keep this going. Can you go at your own pace and get out of synchrony with me? You can just go really fast, you can go really slow. And you hear this noise in the classroom, right? That's non-synchronized firing. And now can we have some synchronized firing? Everybody back with me? All right. So before the trial starts, we have unsynchronized firing. Can you become unsynchronized with me? This is what the neural activity sounds like. Okay, it's a big mess auditorily. Then on comes that crazy green circle inside of all those blue squares, and all of a sudden, a portion of the brain starts firing synchronously. And we can begin to measure when that happens in time relative to when the animal is making an eye movement. Who's following that? Is that working for us? So the dependent variable, the thing that we're measuring, is not going to be just the number of active potentials, but how synchronously are these neurons firing? Okay? And it turns out that we do get measurable synchrony whenever you see on that screen, hey, there's my green circle, right? That's my target. We get some synchronized firing. So we can then ask some questions. Well, where does the synchronized firing occur? Does it happen all over the brain? Does it happen maybe in the parietal lobe? Does it happen in the frontal lobe? Where does the synchrony occur? That's one of the questions that these folks are trying to get to. Okay, who's following that so far? Questions on any of that? Well, yeah, please.
For the eye movement, about how they measure how if like they're looking at the correct thing, not to injure or anything like that, but how do they know that they're actually focusing on the correct answer, or they're just having to look in that direction at the right time?
That's a really good point. Okay, so it's a question of how do we know whether the animal is just responding randomly or not? Okay, so the first thing is when you're the MIT animal in this particular setup, they're bouncing an infrared beam off of your eyes, and they know how your eye is positioned relative to where the stimuli are on the screen. So, for example, the trial won't even start until the animal is fixating the central point. If the animal is fixating in the wrong spot, they won't even start the trial. So the animal has to be on that central dot with respect to their vision. Also, if the animal moves its eyes prematurely, the trial aborts. And when the trial aborts, they get put into a minor timeout for maybe like a second or so. And during that time, they can't earn a juice reward. This animal earns its living by getting juice inside of this experiment. Okay, so now your next question was, well, maybe they went up here to the correct spot, but maybe they were doing that randomly. Well, they only get reinforced for holding that position for a certain amount of time. And we also know that there's 1, 2, 3, 4 different quadrants. And if they were responding randomly, the percent correct would be 25% correct, right? Because if you're just guessing randomly, you'd be at 25%. They could show you in other data that these animals are right more than 90% of the time, whereas baseline performance would have been 25% of the time. But it's an excellent question, right? Okay, really good. Okay, so let's go back to our neural synchrony. Okay, so now that we've got the behavior down, we know that we're looking at synchronized neural firing. Here's what I'd like to call your attention to. For the students in this class, items C and D are going to be more important for us than these up here. These are actually the same kinds of graphs. They've just been normalized. I think they're easier to interpret over here. Okay, so let's take a look at some of these data. And what you might see here is in the pop-out task, this was the feature. This is when you have the green circle amidst all those blue squares. Really, really easy to see. What you can see is that before the animal makes a motor response, in this case an eye movement, we get synchronized neural firing happening especially here in the lateral intraparietal lobe. That's going to be way up top here. Okay? So we can think of that synchrony occurring first, and then much later in the ball game, here's our ball game, and when I say much later, I'm talking about 200 milliseconds. In neuroscience, 200 milliseconds can be a long time. We're getting synchronized firing occurring now maybe 200 milliseconds later in the frontal lobe. Okay? So the first synchrony is occurring here in the parietal lobe, and later on, measurably later on, the synchrony is showing up here in the frontal lobe. First, who's following that? Okay, so there's a sequence going on here, and that's the case when we have the pop-out task. That's the case when we have the one that's really easy to see. It's maybe the green circle among blue squares, essentially. Okay? And what we'll do is we'll draw this search task such that we can see that we have the parietal lobe starting here, and I'm going to drop a little arrow, and we're getting now some synchrony. This is my synchronized neural firing. It starts in the parietal lobe. And then later on, we can observe that same firing here in the frontal lobe. but it starts here and it ends here. Okay, so we'll go, and this is for the pop-out task. Okay, and it goes essentially from the top, from the parietal lobe that way. Okay, this is the search. Yes, please.
When you say synchrony, it means the synchrony within the 25 or 15 neurons in the NIT, right?
Yeah, and so let's make sure we all understand what we mean by synchrony. So let's do it this way. Let's say that the back of the room here, folks who are in maybe the last couple of rows, we're going to put you a little bit further back, a little bit more posteriorly in the brain. You're going to be the more posterior neurons, and then the folks who are in the front of the room, we're going to make you the more anterior neurons, okay? And so what we'd like you to do is we'll start out by getting this unsynchronized firing, and then we'll have this group start first synchronizing with me, and then a few tens of milliseconds later, we'll play that by maybe several seconds later, we'll have the front of the room synchronize, okay? So let's all get out of synchrony first. Here's my rate, okay? We're all out of synchrony, okay? Now, folks who are in the backish portions of the room, can you begin synchronizing with me while the other folks remain? Okay, good. So I'm getting some noise, but I'm also getting some synchrony. The posterior neurons, which are the ones in the parietal lobe, are synchronized with me. Okay, and now all of a sudden these guys catch up, and now the whole room is with me. But the sequence was them first, these folks second. Okay, so there was internal synchrony within the parietal lobe, and that gets picked up here, Harshita. Maybe if you'll bear with me, I'll call it, 150 milliseconds or something like that. And then 100 milliseconds later or thereabouts, we've got this going on 100 or 200 milliseconds later. It winds up more anteriorly. Okay? All right, so we start over here and we wind up over that way. Okay? And that's the arrow is going in that direction. And that was the pattern of neural activity that was going on when you were doing that one kind of search task where you had the green circle amidst the blue distractors. Okay? But then you also had a different kind of search task, and that was the more difficult one. Now you've got that green circle, but it's amidst all these green squares, and that's what they're calling the visual search task, what we call the conjunction. Okay? That's going to be over here, and let's see if we can see what's going on here. First of all, the synchrony is starting out at 0, and then the synchrony begins to ramp up. But what you might notice is that the red line, which is the frontal lobe, is coming on board a little bit earlier than the blue line is. The blue line is displaced a little bit that way relative to the red line. How many people are seeing that? Another way of saying that is in this graph, it's blue first, red second. In the neighboring graph, it's red first, blue second. The pattern reverses. Okay? All right, so that's kind of interesting, and I'll draw that in a different way up here. So now when we have the search task, I'll be right with you, Becca. When we have the search task, we're now starting in the front, and later on, we can show that synchrony occurring in the parietal lobe, but the sequence in this case is starting frontally and moving parietally, and we see that by the reversal of these two trends. Okay? So I wonder if we can do that one more time, just to make sure it's really clear. I'll start out, and everybody will be noisy, and then we'll ask the folks in the front to synchronize with me first, the folks in the back to synchronize with me second. So here's my pattern. Everybody just a bunch of noise. Okay? Now we've got one of those crazy tasks where it's a green circle amidst green squares and blue squares. That's really hard. And all of a sudden, the frontal lobe picks it up, but the back of the room doesn't pick it up. They're still noisy. And then everybody's got it. All right? All right. So now we start out with noise, then we get a little bit of synchrony in one region, it spreads to the next region. There's a spread of neuronal synchrony. Okay? And we get that going on over here. Okay? All right. How many people are following that? So you probably didn't know when you're upstairs doing that, and for the other folks in our class, when you were taking a look over their shoulders, you probably didn't know that we had these measurably different patterns of neural activity going on in the brain, but that appears to be what was happening. Becca had a question?
Yeah. I actually have two questions. First, I'm not exactly clear on what the two vertical lines are expressing. So I know like the first one is a saccade, right?
Okay.
Is the
Actually,
0 also a saccade, or is that?
Yeah, I think that's going to be a saccade. That's probably something like the stimulus onset.
Okay.
Okay, so the stimuli comes on, and then at some later point you have to make a saccade. Okay? All right.
And then my other question was, is there a reason why in D, in the search task, this activity is a little bit more delayed across all.
Yeah, right. So that's very interesting that the neural synchrony is delayed a little bit more. That's very astute. Thank you, Becca. A little bit more delayed in the search task, which is the conjunction task, than it is in the pop-out task. How many people see Becca's point? That here's the time axis, and we get some synchrony rising up out of the noise, relatively early-ish for pop-out and relatively late-ish for search. How many people are seeing that? Let's go back to the data that we collected on ourselves. Okay? And essentially we're seeing that here. We're seeing there's a huge delay in picking up the conjunction, which is the visual search, and the feature stuff, which is what they call pop-out, is happening much sooner. So does that work for you? So there's this nice tie between the behavior and what's going on neurologically. Right? And you picked it out without my prompting you, so thank you very much for making that point. There's one last point. We have about 4 minutes left that I'd like to try to make. And this is, I think, a really, really interesting point. Okay? We'll deal with this graph, okay? And we're going to deal with two different rhythms here. So earlier, the group got to be really good in synchronizing with me. How many people have heard of this phraseology before? Quarter notes versus eighth notes? Has anybody heard of that before? If maybe you've taken some music class. So I was tapping out a pattern that might be considered roughly a quarter note pattern. Okay? And so I wonder if we can have everybody synchronize with me in quarter notes. Okay? If you know what eighth notes are, can you play eighth notes? Can you clap in eighth notes? Okay? Okay? Now can I have the back half of the room stay with me in quarter notes? Can I have the front half of the room go up to eighth notes? Okay, really good. Why don't we stop there? Okay, so we have these two different kinds of frequencies. One is twice as fast as the other. One's a relatively high frequency in time. Those are the eighth notes. One's a much lower frequency in time. Those are the quarter notes. Who's okay with that? Okay, so what's really interesting about this is that the neuroscientists at MIT found that not only is there a difference between search and pop-out in which area is occurring first, but they also occur at different frequencies. That is, on the pop-out trials, we actually got relatively high frequency synchronized firing, and that's shown here by the high frequency line that I've drawn. That's essentially that we're synchronizing in eighth notes. Okay? On the other hand, when we've got a search task going on, then we have neural synchrony, and neural synchrony is now going frontally to parietally, but now we have a much lower rate that's akin to quarter notes. Okay? So not only do we have different sequences, but they're happening in different frequency ranges, and for simplicity here we're calling them eighth notes versus quarter notes. So what's really nice is the same neurons are firing in synchrony in different sequences, but also in different rhythms. Okay? And those rhythms seem to be carrying information about whether this was a pop-out task or whether this was a search task. Who's following that? Does that work for us? And the way that we see that in this graph is here we have frequencies. These are the lower frequencies, and these are the higher frequencies. Okay? And these lowish kinds of frequencies, we have a lot of activity when we're engaged in the search task, and then here we're having a lot of activity when we're engaged in the pop-out task. Okay? So you get this nice fluctuation of which is more active, and so the frequency of firing is telling us basically which kind of task we've got there. How many people are following that? Yeah? Okay, real good. I'm just going to put on the word search so that people who are taking this item down know that this is the one that's search. It's the lower frequency. And I'll also put here parenthetically conjunction. This is the conjunction search. Okay, really good. Well, I'd like to thank everybody for participating in our study. Really glad that you could do that, and I'm really glad that you could hear a little bit about the neuroscience. My students, just a reminder that we do have our exam on Wednesday, so we'll look forward to having you come in and knock that one down just like you've been doing well on the other ones. And then on Friday, we'll be talking about one more article, and this is one that was published by me and my students. Okay? All right. Thanks for a great session, and I'll see some of you Tuesday. I'll see all of you here on Wednesday. Many thanks to our visitors. Glad you were with us. I'll turn up the lights in case that makes this a little bit easier. Dr. Kennedy, come on in, please.
Oh, gracias, gracias.
All right. I'm just going to get my stuff out of your way. Yep, yep. More student presentations today?
No, we are doing reflections on the civil rights movement this week. Looking back to see did it work? Did it not?
Okay.
Where are we now?
All right. All right. Thank you. Thanks for coming.
I don't think
Well, thank you so much.
Thanks for coming in. Yeah. Okay, great. Great to see you.
Yes, I did, and everything is saved.